Okay. Oh, great. Well, thank you, of course, to the INI. This has been wonderful, and program co-organizers who we've developed a really good collegial friendship with, and it's been really fun. I thought I'd give you a short synopsis from my perspective about tracking movements, and so um, I thought that this would be fun to share with you. Of course, tracking dynamics is a huge problem across the spectrum, from a range of people talking about molecules and elephants and birds and fish, and this has been very cool. But why? You know, what are the challenges that we face? And of course, the first one is non-uniform shapes. You know, on the very far left over here are microtubules. We have cells, worms, fish, all sorts of a variety of things. We also have complexities in dynamics. Worms like to interface with one another, cross over each other. Cells like to do the same, such that it's very difficult to tell this is an individual cell or a group of cells. And of course, we have large scales that we've seen. We've seen flocking bird populations. We've seen lots of cell populations coming together in a dish. And so tracking becomes going from a single cell or a single worm to being able to identify different shapes, complex dynamics, and large scale. And it's been so much fun to talk with people across the disciplines. I'll give you a couple of examples. So after the first workshop, I started to look up what are the common tracking tools that people are using, because we started sharing some information from different communities about what tracking tools are you using. And I came across this paper, 28 free animal tracking software packages, none of which I've heard about, but were related to animal tracking. I then looked on the cellular side and found almost an equal number of packages available that were tracking cells or algorithms that were written. And I thought, oh, this is great. This is wonderful information. I can share this and we can discuss. The problem is I've never heard of any of these packages. I've never used them. I even looked at my own home university, and Pei-Jun Lang has written supposedly a very beautiful cell tracking algorithm I don't know anything about, and I don't know the lab that she's from. Now she's gone on and doing a postdoc at UPenn. And I would like to ask Philip or Helen whether they know Martin Hailstone from Oxford, who's written supposedly a very beautiful cell tracking package, but he is now at Nottingham. So it's unclear if we can coordinate these packages or distribute them, and what they have or what really their striking features are that we can share with one another when people distribute and we don't know anything very much about these packages that people are writing, and they're not commercialized, or they're in the literature but might be in different journals than what we really are used to reading. So that's the beauty of this program. It brought together people from various disciplines, from molecules to mammals, and it opened the discussion to who's using what packages, how they may be applied to different scales. And I just wanted to give you a couple of examples. This is a common shared problem, non-uniform shapes. This is C. elegans, very common model system in biology. And on the other side, so for more active work community has put a lot of time and effort into this. They have skeletonalizing algorithms and these may have extended use. So after getting folks discussing between these two communities, it was clear that the worm people are more ahead of the tubulin folks. They have started to develop things back in the mid-2000s with the parallel worm tracker. There's now a worm track. It's a plugin available on a very common free source platform called ImageJ. And so there's even means within the worm community to share worm behavior data and these programs, which is wonderful. It's lacked in the tubulin community, in the cell biology community, really, to some extent. It has even gone further to simplify it for classroom use, which I thought was beautiful, because now teachers in middle schools can set up a worm experiment with a very low-cost camera, run the data, and now they can import those movies into ImageJ and analyze them with the WormTrack plugin. So we've gone from a complete sort of segmentation methods, which are the basis, the mathematical basis for these methods. People brought together at the INI in these different communities from cell molecular biology and development, and it led to an exchange of methods, identified some new challenges, and included classroom education, which I thought was really cool. Now, the new challenges that emerged from these discussions with folks here during the workshops and extended were that now, of course, people are interested in watching lots and lots of tubulin dynamics at the same time. So there are emerging methods in neural networks and deep learning to handle large-scale data sets. There's now a deep worm tracker where they combine synthetic data with real data and apply neural network modeling. And of course, we heard a talk from Simon Martina Perez about deep learning applied to cell behaviors. So this was just one example where tracking programs that are out there in the field appear to be uncoordinated when these communities don't read across journals, anything from computer vision to sensors, etc. But this program at the INA brought people together across disciplines, and the community came together. And I think we're going to continue discussions about what are the most useful algorithms for segmenting something that's elongated like a worm. How can it be applied to intracellular dynamics? And it's a really nice example of a crosstalk across disciplines. Okay. I mentioned the second, one of the other challenges in tracking is complex dynamics. Cells can pass over one another. Birds can cross over their trajectories. And we heard some really cool talks about observing large groups of animals with drones and computer vision, especially from one of our primary speakers, Ian Cousin, and many of his former students from the Center for Advanced Study of Collective Behavior. Now, this was really cool. It stimulated a lot of discussion about GPS tracking of animals. What I thought was really neat was they developed fake animals as drones to both observe and integrate into the real population. And this, of course, was really fun to hear about. And you can imagine that it excited the cell and developmental community now with looking at something that's at a larger scale. And what it stimulated a discussion between several people where, can we develop an implantable fake cell into the adult or embryo that we can control and observe, not only developing a drone for tracking, but implanting this drone within the population. And so this was a really fun set of discussions. I went back to my home university, and I learned that there are folks that are developing self-propelled toroidal robots. And this I thought was really neat. This is a little robot with a biomimetic membrane such that it can be implanted into the human body. And the idea here is that they're going to use this within perhaps the gut to do gut observations and to lead things from one position to another. So it started the discussion as, can we scale some of these ideas of GPS drone-like devices from mammals down to cells or embryos? And so I think this was a really lively discussion that stimulated a lot of curiosity and potentially new avenues to explore. So what I was able to do here during this program was bring together people at the animal dynamics and show how mathematics is at the interface of this, and perhaps mathematics blended with bioengineering approaches. So now we have these couple of areas coming together, but the big cross talk was between the animal movement folks and developmental biology to identify these methods, new challenges, and perhaps can we get this into the classroom? So I wanted to discuss that I really, and the program really helped to coordinate tracking tools at multiple scales. It's clear that even if things are published in different journals and there are lots of different tracking methods, unless people come together to discuss and to share information in person about these tools, that just going to the journals isn't enough to be able to provide a crosstalk so that we can coordinate these tracking tools across disciplines. It also stimulated new ideas where mathematics is really at the basis of both the tracking methods, their details, and these new ideas about blending with bioengineering such that we can take GPS and scale it down to cells and embryos, etc. So I thought that this is really a place to stop and lead to more discussion about what the INI helped us to accomplish and where we can potentially go from here.